Bacterial necrosis of mango in southern Spain, a disease caused by Pseudomonas syringae PV syringae. Authors Francisco M. Zacorla, Juan A. Torres, Laura Olelia, Alejandro Perez Garcia, Jose M. Fari, and Antonio D. Vicente. Mango trees are a tropical fruit tree that is native to India and Southeast Asia. They are a popular tree in many parts of the world and are grown for their delicious fruit. Unfortunately, mango trees are susceptible to a number of diseases, including one caused by Bacterium Pseudomonas syringae by PV syringae. This disease is characterized by leaf spots, defoliation, and fruit rot. It can be spread by wind, rain, and insect, and can cause significant damage to mango trees. To prevent the spread of this disease, it is important to practice good sanitation and crop rotation, as well as to use fungicides and other treatments. For the materials and methods, for the study area, commercial orchards in Malaga, Spain were studied at different locations near the Mediterranean coast, and at a cultivar collection in the experimental station La Mayora of Consejo Superior de Investigaciones Científicas in Algarrobo. Weather was monitored at the meteorological station at La Mayora. For the isolation, to isolate the causal agent of apical necrosis, small pieces of affected leaves, buds, and other tissues were cut from the edge of a recent necrotic lesion, placed in sterilized plastic bags, transported to a laboratory, and processed on the day of sampling. The samples were processed by two methods, which is a portion was surface disinfected by immersion in a sterilized solution of 0.1, and for the identification and characterization of bacterial strains, the bacterial isolates were identified according to the biochemical and psychological test. All inoculated media were incubated at 22 degrees Celsius for 3 to 5 days. Duplicate tubes or plates were run for each test, and tests were repeated at least twice. So, as you can see, this is the example of affected by typical symptoms of bacterial apical necrosis on mango trees. For the picture A, this is tree with typical necrosis affecting most flower panicles, and for the picture B, a tree presenting a stronger attack with the generalized blast. Mango buds, leaves, and stems are susceptible to infections. Symptoms include necrosis of. For the picture C, vegetative and flower buds, and for the picture D, panicles and necrotic lesions frequently extend to the leaf, petioles, and stem. Lesions on leaves start as intervenal necrotic spots and later. For the picture E, they coalesce, become black, and raise slightly. And for the, as you can see, for the picture F, infected dormant buds generally show the initial symptoms. And for the picture G, a detail of an affected bud is observed from an axial cut. And for the last one is when buds were experimentally in inoculated with strains, the Pseudomonas syringae, PV syringae, typical symptoms were reproduced, and generally buds necrosis extended towards stem and leaf petioles. So for the next one, to test the pathogenicity of P. syringae, PV syringae isolates on mango, two experiments were carried out beginning in March 1994 and January 1996. Buds of 2 years old mango plants growing in pots were inoculated with 10 of a 3 times 107 minus CFU ml bacterial suspension in phospholate buffer, and the inoculum was forced into the bud with microsyringe. So for the first experiment, bacterial suspension of 12 strains of Pseudomonas syringae PV syringae, and for the Second experiment, 6 mango plants growing in pots were subdivided into the 3 groups of 22 groups were maintained under field condition in 2 different sites with different wind exposures at La Mayore, and the third was kept in plastic greenhouse. So for each of the 4 buds of each group was inoculated with one of the 4 P. syringae PV syringae strains. And to confirm the presence of P. syringae in inoculated tissue, resolution was carried out from the plant buds showing necrotic symptoms after 3 or 6 weeks. Each bud was surface disinfected with 70% in ethanol, cut into small pieces and homogenized in lab blender for 3 minutes with sterile phosphate buffer. For the result, bacterial isolation, identification, and characterization. The characteristic of the P. syringae strains isolated from deceased mango trees were similar to those of P. syringae strains isolated from healthy mango trees. All of the isolates from asymptomatic and diseased mango tissue were identified as P. syringae, PV syringae. Nearly all the isolates had nucleation activity at -5 degrees Celsius, and all the strain tested were also be able to produce syringomycin and syringopeptine, as demonstrated by the inhibition of G. candidum and B. megatherium, and for the last one is R. palaeminae. 2 pathogenicity experiments were carried out under the different environmental conditions. In the first test, performed under field conditions during the spring, a higher proportion of inoculated buds remained healthy up to 35 days inoculation, 80.6 degree compared with the second test, which was carried out during winter, 18.7%, and also performed under field conditions. This second period was characterized by lower temperature and more abundant rainfall. Typical necrotic symptoms were observed in the most of inoculated buds after 3 and 6 weeks, and the first symptoms were observed 7 days after inoculation. In the buffer inoculated control, no necrotic lesion around the inoculation sites were observed after 6 weeks. In a parallel assay carried out in the plastic greenhouse, the symptoms development in inoculated bud was lower, 31.2 after 3 weeks and 43.8% after 6 weeks, than in a field condition, 71.9% after 3 weeks and 81.3% after 6 weeks. No differences in symptom development were observed between different Pseudomonas syringae, PV syringae strains. Bacterial strains were re-isolated from the inoculated buds that developed typical symptoms. Re-isolation yielded fluorescent bacteria with characteristics similar to the inoculated isolates. The presence of P. syringae, PV syringae was confirmed in 92.5% of experimentally inoculated mango buds, showing apical necrosis symptoms 6 weeks after inoculation. Furthermore, P. syringae cells were present in high numbers in bud tissues, 107 to 108 cf gram fresh weight that developed symptoms. For the discussion, the role of P. syringae strains in the induction of bacterial apical necrosis mangoes was demonstrated by the production of typical necrosis following inoculation of different isolates into buds of young mango plants and subsequent re-isolation of P. syringae, PV syringae from affected buds. 92.5% symptom development in inoculated buds was strongly influenced by environmental conditions. When the temperature was relatively low and the weather was rainy and wet, there was a higher proportion of buds that became symptomatic. The high levels of P. syringae observed in diseased trees could also be explained by the bacterial growth on infected buds coincident with the disease development. because infested bites were incorporated in bulk samples. However, some trees that were healthy at the beginning of experiment and were not initially colonized by P. syringae became infected in spring, developing disease symptoms in the following autumn. In this way, it has been reported that the establishment of large epiphytic population sizes of PV syringae is necessary for the likelihood of occurrence of bacterial brown spot of snap pivia and symptoms of different diseases caused by pathovars of P. syringae do not appear until some leaves or leaflets are found to harbor a relatively high P. syringae population. Our results support the view that syringae is a weak pathogen causing only when the host is stressed. And for my references for what I have presented, and that would be all. Thank you.